Thank you very much for the introduction, for the invitation. So our talk today will be about gold nanoparticle conjugate for live cells and in situ gold immunolabeling. So my name is Zuber, so I'm located in Strasbourg. So if you want to contact me, my email address is here, so zuber@unistra.fr. And my background is not really much in EM field, so I was just developing nucleic acid delivery system, and so I'm more interested in. I was mostly interested in developing system for the delivery of macromolecules inside cells, and so. But you will see it's really much connected to what we are going to talk about. So the context of our work is advanced in cryo-electron microscopy. So basically, this is an image from Mahamid that has been published in Science in 2016. So basically, what you can do right now is you can just froze living cells. You can just have them in cryo in vitro ices. You can carve them, and then you can analyze them. So this is very interesting. For us, because of course you have the opportunity to see everything without any chemical damage to the specimen. So this is very important. So if you have you can see ribosomes, you can see a lot of things, you can reconstruct things. Actually, we were working with Patrick Schulz on here at the EGBNC, so close to our lab, and so they were much more interested in seeing transcription factor, the transcription system into the nucleus. And so the main issue with the nucleus is that it's pretty dense. And so here the idea is to use contrasting agents, and most of the contrasting agents, if you want to use for electron microscopy, gold particles are the one which are the more dense. And so they should be quite easy, quite helpful to find anything into the nucleus, and then to try to reconstruct and to find electronic clouds and to try to reconstruct things within the nucleus. So we have two issues. So the cryogenic state should be maintained. So this is for sure. So we cannot do any immunostaining after cryogenic fixations. And the main issue is to make the gold particles to get inside the membrane. So that's my objective, my specialty, to put the cells, materials inside the cells. So to use gold particles or to make them work into living cells, so the questions were, can we deliver gold particles inside living cells? And then what is the behavior of gold particles inside living cells? So that was the first questions that we wanted to ask. And this is just the theory. So it's just like a physical scheme. So you just see the diffusion ability here of exogenous protein after microinjections into mammalian cells. So this is the D cytosols to D water is the diffusion coefficients. And so we plot here plot the diffusion of the materials versus the hydrodynamic radius. And of course, when you can see that the larger the particles is, the less they diffuse inside the cells. So this is very, very common. And you can see here antibodies here, they diffuse, but not so well inside the cytosol. And then you have materials that's not diffuse at all, but as they should diffuse more. It seems that they stick much more strongly to materials or to cytoskeletons than inside the cells. So you have some behaviors that are very much different. And in regard to gold particles, so they should diffuse, because usually if you have particles around 2 nanometers here, they should diffuse well. They should diffuse up to 6 nanometers. But what is also known for gold particles is that when they have diameter that are above 2 nanometers, you have proteins that bind around the particles, and you have a protein clone around, which is more or less dynamic. And of course, it increased the apparent sizes. So you can have particles that are 2 nanometers, but when you mix them in presence of proteins, on the cytosols contain a lot of protein, you increase the size dramatically. And this has an impact on the diffusions. So basically, at first experiment, we just try, and I just want you to show you what's happened when we conjugate large, so 4.5 nanometers gold particles. So you can see them like this. So they are made by citric acid made. So this is kind of common gold particles. So here we just equip them with peptides that are nuclear localization signals. So basically, if you put them inside the cell, they should get through the nuclear pore inside the nucleus. So we just made gold particles. We conjugate and we linked peptide using gold thiol conjugations, coordinations. And we also put some polyethylene glycol to reduce the unspecific binding. So we just made those gold particles with the peptides equipped with nuclear localization signal and with polyethylene glycol. And then we just put those cells, we electroporate them in presence of cells. We just waited for 24 hours. And then we fix the cells here. We amplify the gold particles with silver. So this is pretty much known for people in the electron microscopy field. And then we just over-amplify to be able to see them just by optical microscopy. So we have done pretty much a lot of experiments like this. So this is the reason why I just want to emphasize on this. So this is the result we obtain with the gold particles that are 4.5 nanometers. So what you can see here is that some gold particles get inside the nucleus, but this is a dead cells. And most of the cells, they do not accumulate the gold particles. So there is several hypotheses that we can make with those gold particles, that either the pore, they did not enter through the pore here, or they do not diffuse, or either the gold sulfur bond did not sustain inside the cells. So that was 3 hypotheses, but we also, so these low diffusions, we can make smaller particles. So to work with the gold sulfur bond, we can also use other type of gold particles. So this is what we've done. And we developed optimized water-soluble gold particles for bioconjugation. So we made gold by coating them with ligands. And so this is the kind of ligand that were very helpful in our case. So basically, it's also a classical way of making particles. So we just put salt, gold salt with this ligand, we reduce, and then we obtain gold particles that are quite homogeneous and with size that are quite easily to analyze by polyacrylamide gels. We found out that this molecule is very interesting because during the reductions, you have this nitro group, part of the ligand here, this nitro group transformed into amine. And this was quite helpful because you have here an amplifier that diminished the unspecific absorptions. We also analyzed by vitro size. Of course, you can see very small particles that are very homogeneous. And the thing is, so then after we try to modify them, so this is just like standard chemistry, and this is very nice because we can do this chemistry in water, so at pH 7.5. You just take a peptide or something which has a thiol, and you can equip those particles with peptide that are functionalized. Here we use the same NLS peptide. And the nice thing as well is that we can very much analyze the number of peptides that are linked to the gold particles just by looking at the retardations of the particles within the shells. The nice thing is here we can almost count the number of peptides that are within the particles, almost. So this is very nice. We can also have we have a batch control, which is very nice as well for the gold particles. And then we just electroporate them inside the cell. So we did the same exactly. And this is to compare the ligands that remain onto the surface, because we cannot substitute all the ligands with the peptide. So this is where you have this amine carboxylate remaining ligand on the gold. And here we compare to thiobenzoate here, which has only the carboxylate. When you electroporate them inside the cells, in HFF cells, and if you wait 18 hours later, you can see that those particles, they seems to stick much less to cytoskeleton or to materials that are within the cell. So they are quite interesting to use for further experiments. Of course, 1.4 nanometers gold particles is kind of very small if you want to look at them directly. And we try to make particles that are of larger size. If we change the solvatations during the synthesis, we can make particles of much larger size. So this is important. We can play with the size of the gold particles. And we wish to have particles that are a little bit above 2 nanometers. And the main issue about those gold particles of diameters above 2 nanometers is that they start to bind proteins and biocomponents. So we just took those gold particles, we just choose those 2 nanometers gold particles, and we did the same with functionalization with NLS. And we also functionalized them with polyethylene glycol to diminish as well unspecific binding. So again, we did the same. We conjugate, so this is the gold particles, 2 nanometers alone. We conjugate with a small peptide here, and then we just modify the ratio between the PEG and the NLS. And you can also try to see that you can follow them pretty much easily. To facilitate the analysis, we also homogenize the cell parameters. So we did some cell cycle synchronizations with thymidine block. So we block the cells here at this area with thymidine, and we just did the electroporation with the gold particles when we release the thymidine block. So the replication did not, the division occur only after 12 hours. So this is, I just want to mention this. So, and then we just try to see whether the gold particles with the NLS, where do they go after 6.5 hours following electroporations in those synchronized cells. So those cells do not get into divisions during this time. So you can see that when you have the gold particles with the peptide, they do not enter they go inside the cytosol, but they do not enter the nucleus. The PEG is even more excluded. And if you increase the number of NLS that bind to the surface of the gold, you can see that you have more and more faster accumulations of the gold particles inside the nucleus. Of course, if you put too much NLS, NLS is also charged, so it can also bind to the surface of the cells, and you can also have kind of accumulations inside the lysosome. So this is kind of interesting. So we took those gold particles with NLS, and we did see movies to see whether we can see them inside the cells and inside the nucleus. So we just high pressures, and that's the work of Patrick Schulz's teams. We electroporate these inside the cells, and then we just did some high pressure cryo freezing sections. So this is the ribbon. We just have some cells here. It's a little bit wrinkled, but this is normal. So this is the nucleus here. This is the nucleoplasm. On here, you have the nuclear envelope here. We zoom it in here. If you zoom it here, you can see very barely, but you can see gold particles within the nucleus. And so this is, we were kind of excited about this. We confirmed that this is not any crap or any noise due to gold particles or to other things. So when we did radiolysis of the slice in the nucleoplasm, so if you remove the water and if you just radiolyze the organic place, only the gold particles remain, and you can really see them within the slice. So this is very important because you can see that those gold particles, we can functionalize them. The NLS seems to remain bound to the gold, and we can also see them without any silver staining. And so this is kind of very interesting. Of course, larger one will be a little bit better, but for the moment, this is the one we have. The next level we went is, well, can we conjugate those gold particles to antibodies? And we try to do pretty much the same. So this has been done with gold particles at 1.4 nanometers. And if you do this, first we just try to link the gold particles at the inch area here. So again, you can follow the reaction by polyacrylamide gel electrophoresis. So what you do, you take the full antibody, you reduce here with TCP the disulfide bond to monothiol, you add the gold particles, and then you just coated the remaining of reactive ligand with polyethylene glycol. And you can really much follow the reactions here. So if you have those gold particles, we analyze them by electron microscopy. I don't want to get too much in details, but you can really much see that they are really much at the inch area here. So basically, we electroporate those antibodies that are conjugated. So I just want to mention that the PEG was mandatory to not have immunostaining ability. So we just did some electroporation of those gold particles conjugate to AGG. We waited for 24 hours and we did some glutarate fixations. Again, we did some silver staining. So this is the result we obtained with antibody that is conjugate to POL2, to an antibody that targets the RNA polymerase II. So you can see that they enter the nucleus, and they seem to stain the nucleus much more like the antibody does it. If you take beta tubulin, you have issue. You don't see the tubulin like they should be. This might be explained by the fact that electroporation destroyed the tubulin network, and you might have some trapping of tubulin in degraded versions. And if you use, so this is with other kind of antibodies, so you can also label nuclear pore. And here we use an antibody that targets PLK1, and you can really much follow the PLK1 during the mitosis. So this has been done on electroporated cells. So inside, so all this immunostaining is done into living cells. So this shows that skull immunolabeling of intracellular compounds in living cells is now possible. We had indeed some issue in relation to the antibody, which is quite large, and we want to reduce the size of the antibody. And the nicest thing to do this is to minimize the size using nanobody. So we first try with nanobody models which target anti-GFP. here we can modify the nanobody quite easily with thiol here. So we use one at initial trial. We modified the thiol at here for encourage this nanobody to gold. But this direct conjugation did not perform well. So we had a lot of loss of binding affinity, and sometimes the conjugate didn't precipitate and did some things that were not working. We found another way to conjugate nanobody to the gold particles is to use mutual recognition peptides. So we just modified this gold particles with a peptide, and we then use self-recognitions of those peptides to link nanobody, which is dimer. So we've done this, and then we just show that this is a classical gold immunolabeling of GFP on H2B2 GFP HeLa. So you can see the histone H2B2 that are fused to GFP. So this is the classical gold immunolabeling by over-amplifications of the silver. And if we do just like mild amplifications, you can really much see the gold particles within the cell nucleus. And then we just did some delivery of the anti-GFP inside living cells. So again, we synchronize the cells. We incubate them for various time, and we just fix them as well with butyraldehyde, and we did some saponin to permeabilize the membrane. So this is the result we obtain with those gold particles that are just conjugated with the peptide. So after 7 hours, they are excluded from the nucleus, 39 hours as well. So the mitosis undergo at this in between 13 and 24 hours. You can pretty much see that those gold particles are very much excluded from the nucleus. And if you use this nanobody that are conjugated to the gold particles, so also in this HeLa that contain H2B2 GFP, you can see that at 7 hours they do not get inside the nucleus, 39 hours they still not get in, but after divisions they start to bind to the chromatin, and they remain inside the nucleus after 24 hours following the time. So this is, and they seem to have the same pattern as GFP H2B2. And if we do some electron microscopy of silver specimens 16 hours following electroporation, so this we just over-amplify the silver enhancement, so you can see that those particles indeed they are mostly excluded from the nucleus. And if you use those gold that are conjugated to the particles, you can see them that are very much within the nucleus at around locations that are in between the chromatin and the heterochromatin. We have redone the experiment here with mild silver enhancements, and you can really much see here, not so much, but you can really see a lot of gold particles within the cells. So this is an magnification again, and you can really see that they are very much on the periphery on the heterochromatin. If we wait a little bit longer, we still have the same things, and you can see that the gold particles you have quite a lot of gold particles within the cells. So I just want to conclude. So the initial question was, can we deliver gold particles into living cells? So yes. The behavior of gold particles is if you coat them with polyethylene glycol, and if you treat them well, they can diffuse inside the cells, and you can equip them with materials that binds to target. So this is extremely interesting. And for the perspective, we would like to further demonstrate that they could be used for cryo-electron microscopy on cryofix cells. Of course, you have different problems, but this is something that we want to solve. You have also other things that are important to verify is here that the cells, we put the immunostaining or the probes within cells that are in motion. So we have to study now the motions of the cells, when to fix them, what to do with this. And of course, the dose on the specific biological interference, if we have antibody that's bound to targets that are inhibiting things, we're going to have a different kind of labeling as well. So, and I just want to thank all my team and my collaborators that has helped me to go at this level. And I thank you very much for your attention. And if you have any questions,
thank you, Guy. Any questions? I don't see any raised hand, but I do have a question, Guy. So when you do the immunolabeling, you electroporate or whatever method you put in these live cells, how do you wash them? Do you worry about noise from extra excess antibody that go in the cells? Do you go through a washing or anything?
So the way we do the electroporation is indeed, for the moment, the electroporation is done in suspensions. So we do the electric pulse here. So we trypsinize the cells. We put the gold particles, we apply the pulse. And then, of course, we just we remove the largest excess of gold particles, pelleting down, and then we just cultivate the cells. And so the particles that are outside, they are removed pretty much. We can wash them very well. So the issue indeed is about the materials that are electroporated inside the cells. So this is something here we cannot wash actually. So this is the main issue. We actually, the fact is, in our case, when we want to label things inside the nucleus, only the things that usually what will happen is if we have a transcription factor here that is synthesized inside the cytosol, the antibody will bind to it, and maybe the transcription factor will get in. So this is much more like the polymerase II is doing it. So basically, only the things that bind to the materials get inside the cells. For the cytosolic materials, it's a little bit more difficult to discriminate between the moment.
So do they end up in lysosome if you wait that some they are?
They do. Yes, we have some accumulations into lysosome as well.
Okay.
But this is actually for C-movie or for cryo-electron microscopy, it's not so bad to have because if they go into lysosome, they concentrate. So here you can see here, I think this is so particles are getting concentrated, so you can pinpoint them much easier as well. So it's also helpful.
Thank you. Yeah.
And the fact is what we believe is actually the concentrations of materials of proteins inside the cells is extremely high. And so if you have materials on antibody with nanomolar affinity, they should bind to at 100% to the materials. The only issue will be if we put too much of gold particles or if the materials that we put in does not bind at 100%.
Okay.
So we will probably have default of, or we cannot label all of them. Hopefully you understand my...
No, you explained very well. Thank you.